Hi, dear students. Welcome back to one more session of genetics. We are quite familiar that DNA plays a very important role in solving various criminal cases. And quite often we use the DNA which has been obtained from body fluids, hair, nails, and etc. To solve various criminal cases. But apart from the DNA of the body fluids, even the skin cells of our body contains DNA. And the use of such DNA could be of very much importance when body fluids or any such visible evidence of DNA is not available. So, touch DNA is otherwise also known as trace DNA, and it is mainly attributed to those samples from any source present in low levels, usually recovered speculatively without the special detection of a body fluid or a particular stain. Biological samples with low levels of DNA not attributed to any specific body fluid are referred to as the trace DNA or the touch DNA. As touching is assumed to be how they were being deposited, we call them as touch DNA. And this could include DNA left by speaking or sweating onto a surface, or a wearer DNA recovered from the fabric of a clothing. And it would also include touch deposits, which will define as material left by touching or handling of a particular substrate. So, by the touching or by the touching of a person, some DNA can be left over. But at the same time, when he is talking or when he is sweating or where like that also, DNA, a little more wearer DNA can be left over on his fabric or clothing which has been found at a crime scene. Now, what do you use this touch DNA for? This touch DNA simply refers that you can collect the skin cells which are subsequently typed for DNA in the exact manner as a body fluid is used using standard laboratory procedures. So once you get a touch DNA, that touch DNA can be used just like you use for the other typing techniques. So you can use the same touch DNA also. But when does this touch DNA become significant? It becomes significant when no other visible body fluids or any other type of visible sources of DNA are not being present in a crime scene, we can further search for a touch DNA at the crime scene. If you go to look onto this is from an article provided in the Forensic Science International Genetics. You can see that a concept of map of potential sources of DNA deposited by touch or handling. It could be cell-free DNA, or it could be fragment associated residual DNA, or sometimes you can see transferred exogenous nucleated cells, or sometimes endogenous nucleated cells, or air nucleate corneocytes. So different types of cells or the DNA associated with it can be left over on the surface of various objects. Now, what are the different types of objects can we use to get this touch DNA? Anything, like it can be firearms, knife handles, weapon handles, fire casings, uh, steering wheel or other vehicle swabs, fingerprints of the victim, ligatures, handcuffs, shoestrings, hand swabs from suspect, cell phone swabs, victim neck swabs, swabs from torn or forcibly removed clothing, airbags, tools, baggies, paper demand notes, clothing items, hats, masks, gloves, glasses, etc. Any of the evidence which has been available at the crime scene and can be used as a source of touch DNA. Now, you mentioned about touch DNA, but how do you get this touch DNA? An important thing when we collect the touch DNA is that everything, even when the personnel who is taking or the technician is taking a sample from a crime scene, his DNA also could be contributed towards in that particular crime scene, because touch DNA is something which has been found to be invisible to our naked eye. So, if you are appointed to collect a touch DNA, make sure that you wear appropriate personal protective equipment, gloves, and everything in order to prevent your DNA to contaminate the crime scene. And once you are protected and you are not contributing your DNA onto the crime scene, we can sample the touch DNA. Now, how can we sample it? The most common method for the optimal collection of cellular material is a swab technique. Here you use sterile cotton swabs on the surface of the object. And to improve the quality of the resulting DNA profiles, a double swab technique is usually being applied. Now, let's just have a look up on the swabbing technique. Anything, any article which has been found on the surface of a crime scene, you can take a swab and you can swab it and take it. When the swabbing method is utilized, the surface of the item is usually rubbed with a wet cotton swab, and then it is being followed by a dry cotton swab in an effort to collect the possible skin cells. And the wet or the dry swabbing method is recommended for hard, non-porous items such as metal, glass, plastic, and it can be easily performed at the crime scene with limited risk of contamination with exogenous DNA. Now, coming to the next method. The next method which we have is a cutting method or the cutting out method. It is a frequently used method in a large number of forensic laboratories, and the area of interest is usually being at the what does it means the usually the soft items which are being left over in the crime scene are being used for the cutting technique. Now, for the cutting technique, now mostly it will be cloth samples. Now, if you're going to consider the cloth samples, from where do you cut and take? Now, if a clothing is left over at the crime scene by the perpetrator, pressure points on the clothing, such as the interior neck of a shirt or the inside headband area of a hat, they are excellent candidates for these sampling methods. And in a sexual assault case in which the victim's clothing has been removed by the perpetrator, areas such as the waistband may contain sufficient cells belonging to the perpetrator to produce a particular profile. So, we have the swabbing technique as well as the cutting technique. And now let's move on. The third method called the scraping method. In the scraping method, the surfaces of the soft or the porous items are either scraped with a sterile scalpel blade in order to get the sample. And yeah. And after that, we also have a tape lift method where adhesive tapes can also be used. So, in the scraping and the tape lift technique, the analysts will mainly focus their sampling to an area of damage or the area where the perpetrator is believed to have had the most contact. And through the use of these sampling methods, an area approximately of the size of an adult's hand can be sampled. So you can scrape or tape lift from almost an area from the adult's hand as the size, an area as the size of an adult's hand. And the scraping of the tape or post-it notes are then placed directly into the extraction tube. And labs use pre-treated tapes and will also process a blank piece of tape along the side of the evidence sample to ensure that no DNA has been introduced via adventitious contamination from the manufacturer. And the scraping and the tape lift methods allow a larger surface area to be sampled as opposed to the cutting method, which only you get only a limited amount of sample. And as the surface area which has been analyzed has been increased, the chance of recovering more skin cells is there, and as a result, the chance of obtaining a DNA profile, a better DNA profile, is there when you use a scraping and a lifting technique. Other than this method, we also have other methods also. Sometimes you might also use vacuum based methods of analyzing uh, DNA samples, but most frequently we have swab techniques, cutting methods, scraping methods, as well as the adhesive methods also. And the adhesive tape method is quick and straightforward, but the DNA extraction is challenging due to the stickiness, rigidity, and the size of the particular tape. Now, whatever be the technique that you use to isolate your DNA, there are certain things which you have to keep in your mind, because every technique has its own limitation. Okay, and since we're dealing with touch DNA, even our DNA sample, if you are a technician, might infiltrate and contaminate the sample of evidence. So, limit access to the crime scene or evidence is needed. And when you are collecting a sample, do not talk over the evidence, and wear personal protective equipment, including the face masks. Change the gloves frequently after handling evidences, and do not touch areas on evidence that may be sampled for DNA. And you should also remember, if you're using a sampling device at one particular site, and if the same sampling device is used at a different site, what happens? Sometimes you might contaminate the samples from either sites with each other. So make sure that you should change your gloves, and the sampling equipment also should be freshly used. And always collect elimination samples from those who have been in contact with the evidence of the with the evidence or the scene, and use disposable fingerprint brushes as well as powder, as I told you earlier, because you might contaminate it if you are using the same brush or the finger powder at two different crime scenes. And clean each crime scene tool coming into contact with the evidence. So these are the basic rules which you should follow when you are handling touch DNA in order to avoid the possible way of contaminations. But we have great stories in which what happens, touch DNA has been used as a tool to solve various crimes. But you should also remember that DNA is something which can move from one place to the other, especially when we are dealing with skin cells. Now, for example, if I borrow a scarf which my friend wore and I bring it somewhere, I can transfer her skin cells to some place that she has never been to, or an object that she has never touched. So, if in a crime scene her shawl happens or a scarf happens to be there, even if she has not come there, her DNA is reported to be there in the crime scene. So, as a result, in some cases, faulty assumptions can also be made based if you just rely only on touch DNA. Of course, touch DNA helps to authenticate the presence of a particular person, but we have to also consider certain other cases when you are relying on touch DNA as an evidence source. And one such faulty conviction due to touch DNA was being reported. It's a case based on this, where a homeless man named Lucas Anderson, he was charged with a capital murder for the death of a Silicon Valley multi-millionaire. But there was no way that Anderson could have committed the murder. At the time of the murder, he was hospitalized and nearly comatose and under the constant medical supervision. But it turned out that a paramedic who treated Anderson were the same ones who responded to the murder scene, and the paramedics accidentally planted Mr. Anderson's DNA at the scene hours after assisting him. And that was a leading example of the risk of a criminal justice system relying only on touch DNA evidence as an infallible proof of someone committing a crime. So, what we have to say is you have to always think wide, out of the box, considering all the evidences rather than relying only on one particular evidence. So, I hope you are clear with this. With this, we come to an end of touch DNA. Thank you.